Thank you all for joining today. I see that we have quite a few people on the call. So I think everybody knows why they're here. We're going to be talking about docking in particular, but we're going to focus on pose prediction and how you can actually predict the bound conformation of things that look quite a bit different from the things that you know about. Okay. We're also going to show a demo of a graphical user interface that we'll be releasing very soon that shows how you can do everything that we are presenting results on today, but in a very simple and easy to use interface with a platform that I think a lot of you are probably very comfortable with and familiar with. So pose prediction. Why is this hard to do? Why is it hard to do with many docking methods? Well, one of the things is that if you think about all of the methods that you've used, for docking in the past, a lot of times you'll find that it's necessary or strongly encouraged to basically supply a template or some guidance to a docking procedure in order to get a result out that you are happy with. We see this as probably the wrong approach. And it turns docking into an exercise in confirmation bias. So you can do it when you have very near analogs that you're docking, but if you have something that's genuinely different, it might not work at all. And you need to understand how frequently you can believe a real prediction that's an actual calculation that didn't require a lot of knob tuning. A pain point for many people during docking or preparing for it is dealing with PDB structures. I could spend 3 days talking about the things that we've encountered over the past few decades dealing with PDB structures, particularly having to do with obtaining sensible ligand structures when processing through PDB files. The other thing that's really challenging for many proteins, you can find lots and lots of structures out there, but because of the nature of crystallography, they're generally not in the same coordinate frame. And so actually aligning the binding sites and then selecting, well, which few out of maybe 100 or even 300, we'll see in a few minutes, do I want to use if I want to use ensemble docking, if I want to look at protein variants. Okay, and macrocycles, which we've been spending quite a bit of time on over the past several years, create additional complexities for docking. Fundamentally, there's the issue of conformational search adequacy, where many methods require specialized procedures instead of just running what you know how to run. The published results that are out there right now, except for our most recent paper, really struggle to succeed even when you're taking a macrocycle out and then trying to redock it into its cognate protein conformation. And so we're going to present results today that show you that this doesn't actually have to be a real problem for a large class of macrocycles. So why is this important? I think most of you probably know these things, but for us, we've worked with a lot of different companies over a lot of different years. So we've seen many applications of sort of the need for really understanding how a protein binds a ligand, how that ligand fits. One is obvious. If you do high throughput screening, you get some positives. You go ahead and do retesting, you do structure verification, resynthesis, and so okay, we have some real hits here. Maybe you don't have crystallography in-house or it's slow for a particular target. You want to know whether the new hits actually bind where you think they do, and whether you can, say, take advantage of structure-activity relationships from previously published data or from patents, and maybe apply that to your new scaffolds and really get the jump on hit to lead and lead optimization then after that. Scaffold replacement is often important where you have structurally interesting ideas, particularly now where a lot of them can come from generative chemistry approaches. Problem with these approaches, right, is you actually have to make these molecules. You don't get to just buy them. And so you don't want to sit there and contemplate, well, which of these 5,000 things should I make? You want to have a way to sensibly choose. And we believe that docking in the way that we'll talk about is a good way to do that. Another common thing now, increasingly, has been macrocyclization of non-macrocyclic leads to try to improve potency and selectivity. But when you're doing this, having a way to actually dock a macrocycle that's a synthetic candidate, understand how it very likely binds, and understand things about its bound strain and so forth can really help. Obviously, lead optimization is something that people care about. Generally speaking, docking and scoring is not how you want to evaluate something, but you might actually need to have good starting poses for synthetic candidates, whether you're doing MMGBSA or FEP plus or something like that. And often this is harder than you think it will be. Okay, we're going to spend just a few minutes on theory and methodology behind Surflex-Dock. Ann and I looked back and asked when was the last time we actually talked about how Surflex-Dock works and why it's different. It was a long time ago. So we want to just remind people why it's a very different method than other things that are out there, either in the open source world or in the commercial world of alternative methods. So one thing that's different is that Surflex-Dock it's old. Okay, so it's a descendant of the hammerhead approach, which we developed when I was at Eris in the early 1990s. It was published in '96. We were using it well before then. And it was funny. Ann and I were just down here actually at 385 Oyster Point Boulevard a couple of months ago. The building's still there. Eris is long gone, but the method still exists now in the form of Surflex, which we introduced in 2003 during the time that we were at UCSF, but also jointly running biopharmics. So what we did was take some of the good ideas from Hammerhead involving things like scoring functions and protomols, and we'll get into some of that in a minute, but then re-implement it into a much cleaner and more effective and more automatic set of methodologies. Hammerhead was largely automatic. Surflex has always been fully automatic in terms of every aspect of the docking workflow. These papers and the related papers around Surflex and Hammerhead have been cited thousands and thousands of times if you look at the collection across all of the methodological papers that we've published. One of the things that was very important in the early part of the 2000s was that the field in general got much more involved in independent validation of methodologies and public benchmarking, public data sets, and so forth. People like Greg Warren, Brian Schwickett, and John Irwin at UCSF, people at various companies and so forth. The folks at Aztecs have been great about this, Marcel and Michael and so forth. And we were involved in this early on. And the thing that happened there was you actually got to see a few methods come to the top in terms of top tier docking approaches. Surflex-Dock has always been in that tier, and there are a couple of others, and they're all commercial. So the ones that generally have fared well in terms of public benchmarking, whether it's been done by academic labs or been done by industrial groups, pharma groups, it really has not been open source approaches. And we're going to sort of pile on that in a little bit as well. Okay, so the answer to what's different about Surflex-Dock is everything. Okay, so in terms of the real guts of it, most Dockers are extraordinarily dependent on their scoring functions, and Surflex-Dock's scoring function is very, very different. It uses completely different functional form than other ones. It's an empirical scoring function. It was trained both on positive data, things where you know the binding mode of a ligand and its affinity, but also negative data. So we actually take advantage of decoys that are synthetically generated computationally in order to estimate things like repulsion. So we end up with a very different looking function, both in terms of the sort of hydrophobic steric term. In green here, we're showing the Surflex-Dock scoring function for hydrophobic contacts. The scoring function is parameterized in terms of PKD, so positive is good. As atoms become distant, these are pairwise potentials, you get 0. As you get together and form a nice little interaction at the molecular surface, you get a positive bump of about 1/10 of a kcal per mole, roughly. And then as you interpenetrate, you get penalized. You can see, though, that compared to a Leonard-Jones potential, the interpenetration term that we have is much more forgiving. So it's quadratic rather than an extremely stiff, sort of infinitely painful clashing term. A lot of dockers end up having to clip a Leonard-Jones style term in order to avoid rejecting close to correct poses. We've solved that problem by inducing a function that works well with variations in protein conformations. On the polar side here, I'm showing you a hydrogen bonding term. I'm not showing you directionality, which is also an important part of hydrogen bonding. In green, we have the Surflex-Dock term, and you can see that it falls off pretty rapidly. So within about one water shell of distance between, say, a proton and a carbonyl oxygen, you're basically down to zero. This is very different than a Coulombic term, which even if it's distance dependent, it falls off very slowly. So the Surflex-Dock scoring function tends not to overweight electrostatic interactions in a way that many other docking approaches do. In the red here, I'm actually showing you a potential that was derived by Tanya Kortem's group using a PMF strategy. And what you find is pretty interesting. So using something that's a completely different approach, statistical physics, you end up with a potential that looks very similar to the Surflex-Dock potential, very different from the sort of force field-based potentials that people have used. There's a good to beat my own chest here, but there's a review that I spent a lot of time on back in 2006 that goes through a number of the different empirical and physics-based scoring functions and compares and contrasts how they are formulated, how they're parameterized, and so we encourage people to look at that paper. So the scoring function is completely different. One other thing that's very different is we actually use a thing called multiple instance machine learning to induce the shapes of these terms. And I don't want to get into the weeds on this too much, but if when you're tuning an empirical scoring function, you're trying to learn the parameters, like what's the right distance for a hydrogen bond, if you take a complex like biotin streptavidin, this is 1-STP from Pat Weber from a long time ago, what you'll find is things that are weird, like this carbonyl oxygen in the oxyanion and anion hole here has an extremely short distance to its partner at 1.65 angstroms. So if you were to statically take a structure like this and say, I want to learn about what a hydrogen bond distance should be, you're actually gonna say, well, sometimes it can be really short. And this is a very high affinity complex here, about 10 to the minus 13.4 in terms of the dissociation constant. So what we have done is say, okay, well, there's actually a manifold of poses for the ligand, all of which approximately equally well fit the electron density. What we're going to listen to in terms of the distances that we should be learning and the functional form that we should be learning is the one that maximizes the value of the scoring function. So there's a lot that we can talk about this, and that's another several hour kind of thing. Bunch of papers that we're putting on here, the citations for them, so you can go look at that. But it turns out if you are able to do this correctly, you're able to induce much sharper terms that make a lot more sense because you're not listening to basically poorly fit ligand positions in crystal structures. And we're going to touch on this again later in the talk. Okay, the next thing that's different is the search methodology. So it's underpinned by force gen conformational search. This is a methodology that we introduced in 2017, and it really changed how we approach the idea of conformational elaboration in docking. So we generate ensembles ahead of time. We refine them in docking, including all atom Cartesian optimization. So if we take a macrocycle like this base inhibitor here from 3DV1, generate 3D and generate conformations using force gen, I'm showing you 182 of the roughly 1,000 conformers generated here, compared with the crystallographic pose. So these are all within an angstrom and a half of the crystallographic conformation, which kind of gives us a lot of shots on goal when we're docking these things into, say, a non-cognate base site. We want to make sure that we have a lot of opportunities to kind of fit and then do some optimization afterwards. So using force gen is a big thing, and it's a different approach than we had started with way back 30 years ago. We've always used what we call protomols to characterize an active site, generating molecular fragments that mimic interactions of a ligand. So you can think of it as an idealized binding site ligand, these little carbonyls and NHs and methanes. What's different now is that instead of really focusing on just the deepest central part of the active site, we adaptively scope a protomol based on a measure of buriedness. And so this is CDK2 with a small inhibitor here. You can see that the protomol covers the inhibitor really well, but it also extends way out into the solvent front around CDK2. We also calculate a notion of what the most buried parts of the binding site are. So the yellow spheres here are marking the deepest part, the orange ones sort of moderately deep, and the pink spheres here are marking the things start to reach towards solvent. So now we're using this calculation of buriedness and the protomol in order to help guide the docking. One other thing that's different about our approach is that we can incorporate prior knowledge both in the search process and in the scoring process. And we introduced this idea back in 2015 with the pink data set. The pink is not cognate, so it was the first cross-docking paper of substantial size in terms of the benchmark. So we use prior known ligands to help generate poses using similarity during the docking process, something you can turn on or off if you want to. We also use known poses of prior bound ligands in the same way that people tend to think of them. So if you're somebody who knows a lot about kinases, and these are kinase inhibitors over here, CDK2, you'll know when you see a hinge binding sort of motif that looks sensible because you've seen a lot of kinase inhibitors before. What we've been able to do is systematize that kind of thought process by using molecular similarity to calculate the similarity of a docked pose of a ligand that you care about with prior known poses of other ligands that you have aligned into the coordinate frame. And we use a statistical physics approach to then adjust the docking scores in order to help us with pose re-ranking. And there are detailed results about this in the paper, which I should mention now, that we have just published with our colleague Himani Tandon at Optibrium. I think this is our first official Optibrium publication, so we're very excited to have gotten that out. This just appeared, it's open access, so we encourage people to go and grab that paper. There are a lot of details that we can't go through during this webinar. Okay, so I'm almost done with the theory part here. I apologize. So automation is the last piece that's different. One of the things that I think a lot of users do is spend a lot of time hand preparing things, hand charging things, hand moving tautomers around, doing a lot of different things to try to get results out of a Docker that they want to see. We have always taken the complete opposite approach and said, no, okay, give us PDB codes. And then at the end, we want a docking ready ensemble. We don't want a human being to think or to worry or to pull their hair out over this process. So Anne's going to show you how to do this. But honestly, so PDB codes go in. I think we're 35 or 38 that went in here. They get processed. We grind the ligands. We get bond orders. We do automated binding site alignment. And then we choose examples of protein binding pockets from this aligned set that cover the conformational variation of the binding site reasonably well, okay, as well as we can. So we're not picking the 5 weirdest structures here. That turns out not to be a good idea. We're trying to cover the 5 most informative structures to tell you a lot about how ligands bind and what kinds of changes they'll induce on the active site. So then when you get a new ligand, the only case you really care about is where you have a ligand where you don't really know how it binds. So all the papers that you read that try to do validation by cognate ligand redocking, people stopped really caring about that who do methods 20 years ago now, at least 15 years ago. So you care about cross-docking. You care about, I got a new ligand, what does it do? All right. So now I'm going to hand it over to Anne, who's going to show you how to actually do this from within our soon-to-be released Pymol GUI. Anne, you can take it away.
Okay, so I believe that you can see my screen. What I'm going to show you today is I have the privilege to display and demonstrate the new biopharmic Surflex-Dock Pymol GUI. We want to thank our colleagues at Optibrium for this excellent and easy to use GUI. And now Surflex-Dock will be available to a much larger group of researchers beyond those who are command line aficionados. So what you're looking at here is that this is the GUI, okay? And I've already, when you open Pymol, you just go up under plugin and it says Surflex-Dock, and I have chosen that. We're on the first tab, project. I've named the project non-cognate temporal ensemble docking, and this is very important. So this example is going to demonstrate the concepts that Ajay just described. And then you just give a folder name and a location. The protein that we're using is CDK2. It is a serine threonine kinase. It represents a large class of therapeutically important drug targets, and there are also hundreds of publicly available structures. So the first thing we need to do is prepare our ligand. So we're going to choose 2XMB. We're going to load the SMILE strings, and we're going to accept the depth of search that is suggested by the GUI. I do want to say this is a geometric level of search, and it is sufficient. You get up to 250 conformers, and it's sufficient for most pose predictions. There are more thorough levels of search, and Ajay is going to discuss those. Okay, so we have generated, this is 2XMB, all right? And let's look, it says that we got 249 conformers, and let's show that ensemble. So there they are, you see, and that makes sense because there's a lot of flexibility in this ligand. Okay, so the next thing we need to do is we need to prepare our proteins. And again, we just go and we choose PDB structures. What I've done here is I have a small subset of old CDK2. These 10 here are old CDK2 structures, and you're going to notice that I'm actually including in the download and preparation the structure of the ligand that we're going to dock. But the only reason I'm doing that is so I can put it in the same coordinate frame and evaluate our docked poses against the crystallographic poses. So you just choose your PDB codes and you import. So what's happening now is you'll see at the bottom that 11 are running. And so we're acquiring the PDB complexes and we're separating the proteins and the ligands. And you'll see them starting to populate over here as they run. So here's our first structure, one CKP, and it's a ligand PVP, and another, and a DM2 and its ligand, and so forth and so on. So one thing I do want to point out while this is finishing is that you'll notice that for many of the complexes that it says that it's a quality ligand. For example, for 1DI8, you'll see that it says quality ligand, and for many of these it says that. But for a couple, it merely says ligand. And I want to point out something that's important here for the automatic processing of these structures is there are many problems with PDB structures. And so if you can avoid using structures that have obvious problems, you should. And so what I want to say here is this PVP ligand that I'm showing, we know for certain that the ligand in the crystal structure does not match the SMILE string associated with that head code. And so we would advise users not to use that structure for anything. And that's also true for 1DM2, where this ligand, the HMD ligand, is under more strain than it should be. And so we would advise not to use that. Okay, so now that we have separated the proteins from the ligands and the waters, and we have physiologically protonated them. What we want to do is we want to align and cluster the proteins, okay? And the default suggestion is only use those that have quality ligands, and yes, do that, okay? So while this alignment is taking place, I want to take a few seconds to discuss PSIM. PSIM is our protein pocket similarity algorithm. Notice I didn't say protein similarity. PSIM does not align proteins based on the carbon backbone. It aligns them in a manner that's akin to eSIM similarity. So the eSIM algorithm is looking at small molecule ligands from the perspective of a protein. The PSIM algorithm is simply the flip of that, where you're looking at the protein binding pocket from the perspective of potential ligands. Okay, so now what we want to do is up here we can select, let's look at the clustered proteins. We can select all of them and then choose to show them. And there they are. Move this down. And you can see the beautiful alignment there of those protein pockets. And again, it is the protein pockets that were aligned, not the ligands. The ligands are transformed. And so what you have, if we were to hide the proteins for a moment. Okay, sorry. Show them all again. Okay, so if I were to hide these proteins, what you will see is just the transformed ligands. Oh, sorry, I'm having a little trouble with my select. Let's see. Okay, so there are the transformed ligands. Okay, so now what we need to do is that we need to choose the representative subset of these proteins that we want to use for the ensemble docking. And so we simply choose K, and in this case, I'm going to say I'd like to use 3 proteins. And so while this is running, what's happening here is that, and the process is already done, is that you're choosing cluster centers, okay? And we'll turn off the other proteins. And we'll look only at these 3 cluster centers. Okay, and then we'll turn off the other ligands as well. Okay, so now what we need to do is that we need to generate protomoles, okay? And what the protomoles are is that we need to, sorry. Okay, there. Okay, so now we want to generate the 3 protomoles for our cluster centers. And again, what the protomoles are is they are idealized ligands that are scoping the binding pocket. And there you see them in the context of the proteins. Okay. All right, so the next step, we now have our protomoles. We've aligned our proteins, and now we want to actually perform the docking. Okay, so what we're going to do is we're going to say, yes, we want to use those 3 protomoles. And it says use the quality ligands only, but because we don't want to cheat, as I said, this was aligned. So we want to specifically eliminate the ligand that we are docking from the known ligands. And then we say, yes, the ligand to dock is 2XMB, our 249 conformer pool. Okay, and we're going to dock at the PGOM level. Okay, while the docking is running, I just want to talk about this example for a second. It's actually from our pink data set. And what the pink data set was a temporal data set where we had gotten all of the structures for many different types of targets. And what we did is we chose proteins from the earliest 25% of the proteins available with the PDB, and we predicted on the future 75%. And so you're seeing here the aligned pockets of the 3 proteins that we're using for the docking. And it's important that you do see the variation in the pocket, because remember, our cluster centers and the protomoles are supposed to represent the pocket variation. Okay, so our docking is done. and let's choose to look at it. So there's our ligand in the pocket. I'm going to show the protein controls, but I'm gonna turn off a couple of the proteins and just leave one on so it's easier to view our docking result. What you can see here is the expected interaction with the hinge binding regions of the protein, this hydrogen bonding network, okay? What you can also see is that there are actually pose families that resulted from this, okay? The first pose family here, you can see, has a very high probability of being right. And if we sort of click through and show the members, you can see some flexibility here. And you see this particular moiety down here all aligned. Let me remove the protein. You can see this particular moiety here all flipped in the same orientation. If we were to look down and look at the second pose family, okay, you would see that moiety is flipped. And we know from X-ray ligand refinement that both of these are accommodated by the X-ray density. And then finally, what we can do is we can ask, how well do our docking results compare to the crystallographic ligand? And you can see the crystallographic ligand is in white, and you can see that those top, the poses in the top pose family comport very nicely with the crystallographic pose. Okay, and with that, I will stop my share and turn things back over to Ajay.
Okay, great. Thank you. Okay, so now that we've seen that you can go literally from PDB codes and a smile string all the way to a docked result, what we want to do is talk about the actual validation of the underlying methods and how well they do in terms of actually predicting poses. All right, so Anne talked about this a little bit, that we introduced the pink data set in 2015, where we had 10 targets, nearly 1,000 ligands. We took the fraction of targets that were available from June 2003 and earlier, okay? This amounts to just a quarter of the complexes that are out there. And we use these to identify proteins and ligands that were known at a particular time point, did fully automatic preparation alignment and ensemble selection, just like Ian showed you for CDK2. Some of these cases had hundreds of structures, but some had quite a few less. We kept the 10 targets that had the largest number of examples for us to test the docking on. The remainder came from after July 2003. The XNB ligand called Y8L that Anne showed you was actually deposited in the PDB almost 10 years after the known structures were. So we're looking at very different scaffolds, new structures, new underlying scaffolding for these ligands. So really challenging in terms of trying to predict poses. What we've done in the current paper is we re-prepared the entire data set with higher quality control. And so now we have 846 test ligands. These are nearly all not macrocycles. I think there's a handful, maybe a dozen that are macrocycles, but they're very diverse, okay? And the targets include things like HIV protease, thrombin, CDK2, MAP kinase. There are the kinds of proteins that were very well studied early on in the pre-2000 period. This time for this paper, we wanted to look at how well we could do on macrocyclic ligands, and we ended up with 13 targets that overlap a little bit actually with the non-macrocyclic target set. What we did was go into the PDB and identify with from the Ligand Expo smile strings all of the macrocycles that exist in terms of being solved in complex with proteins in the entire PDB. We then looked for the corresponding proteins and uniprot codes and found all of the non-macrocyclic ligands that they were bound to. And we constructed a data set where we're going to use the non-macrocyclic ligands over here, macrocycle naive structures, binding to normal small molecules, and try to then generate ensembles, which we did again automatically, just the same as before. So now into these macrocycle naive structures, we want to take all of the macrocycles then that were in many cases actually designed to specifically try to engineer more potent, more selective molecules. But what we wanted to do is dock these macrocyclic ligands into these non-macrocyclic bound structures. Okay. After all of this data set preparation, which again, all automatic, we ended up with 128 ligands that were macrocyclic to test. And obviously they're very different than known molecules, which were none of which were macrocycles. Okay, so I'm just illustrating now here this procedure. We've shown this already, where for CDK2 there were on the order of 38 structures, 33 of which go through the quality checks. You can see these ligands all on top of one another, making a characteristic sort of hinge binding motif against the hinge of CDK2. One of the things I mentioned earlier is that Surflex-Dock takes advantage of these known ligands. Okay, so Anne showed you that in the Pymol GUI that the known ligands help drive both the search and the docking, but also help with re-ranking of pose families in the end. We get 5 variants. This is all done. And then we dock a new ligand. Okay, in this case, deposited much later than these structures were solved. On the right-hand side, I'm showing one of the particularly interesting macrocycle cases. So base 1 has been a very well-studied target. As you can see, there are over 300 non-macrocyclic base ligands that have been designed, tested, co-crystallized, and actually deposited publicly in the PDB. You can see that there's a lot of structural variation in what these things actually look like. More than, I've been doing this a really long time, and I look at these things and I wouldn't immediately guess that they bind the same site, but they do. So we can process through a very large data sets like this and then end up with 5 selected structure variants. The ligands in particular are these 5 over here. So now the game is, we have a base ligand that was deposited as 3DV1. We want to try to predict the pose of this ligand based only on this information from non-macrocycles. So there have been a few papers published out there in the last 5 or so years talking about macrocyclic docking. All of those papers that we can find are talking about pulling a macrocycle out of its cognate site, maybe not even randomizing its coordinates, and then trying to dock it back in. Okay, they don't even work doing that, not well. Okay, if you randomize the coordinates and generate an agnostic conformer, the situation is pretty grim. So even the papers out there that have gone after macrocycle cognate ligand redocking have not reported particularly stellar results. So the temporal split, the bottom line number is that roughly 80% of the time across all the targets, we get the correct pose prediction, either as the top scoring pose family, as Anne showed you for 2XNB, or the second top scoring pose family. So here I'm showing you cumulative histograms for base non-macrocycles, CDK2, HIV protease, thrombin. On the x-axis is RMSD in angstroms for top pose family, top 2, 5, and 10. And you can see that as you include more pose families, you get more and more success. Right here's 2 angstroms RMSD, which is typically considered to be the success threshold in docking. And what I'm doing is circling on each of these graphs the success rate that we see at the 2 angstrom RMSD threshold for the top 2 pose families. So that's in green here. So purple's 1, green is 2, cyan is 5, and goldenrod is 10 pose families. And you really don't get much benefit going beyond beyond 10 pose families. All right, so here you see for CDK2, we're close to 90%. On top 2, for base, it's 80%, and it hovers between sort of 75 and 85, 90% across most of the targets. If we look at the full set of data, all 846 ligands, we end up at about 78% correct with the top 2 families and close to 70% with the top scoring pose family. Now, for those of you who know a lot about the docking validation studies, particularly in the early 2000s, you'll see that these levels of accuracy and success are pretty close to what people found with challenging cognate docking sets. So we're doing as well with docking very different structures, very new structures, in some cases very flexible molecules into naive structures that were bound to very different scaffolds, and we're getting roughly 80% correct across the board here with the top two solutions returned. On the left, just to emphasize, this is the procedure. Randomize the coordinates of the compounds to be docked, completely zero them and regenerate coordinates, build protomols, generate conformer ensembles for each of the conformers, and then dock them using the known poses that Anne showed you, and then I showed you on the previous slide. Build the pose families now using the hints from those known poses, and then evaluate the RMSD. We do that using fully automorph corrected RMSD calculations, so symmetries in the molecules are automatically taken care of. So again, completely automatic procedure, 80% correct. So this didn't surprise us that much. It's a little bit better than we had done in 2015 because of the improvements we've made in conformer search and in binding pocket characterization. One other thing I wanted to point out, I forgot, in the red cumulative histogram here, we're showing the raw success of pure conformer search. So without doing the docking, we're asking whether there exists for each of the 846 ligands, if there exists a conformer that's close to crystallographic. And you can see that 95% of the time, ForceGen is able to produce something, at least one answer that's 1.5 angstroms RMSD or better. So conformer search is not the bottleneck in terms of docking with Surflex-Dock. ForceGen does a very, very good job with non-macrocycles at identifying good conformers. These then go into the algorithm here after. This is the ForceGen search. Okay, and that's what's in the red dotted line here, and then the output is the docking, and those are the other four lines on the graph. So I guess the big news in the paper and in the results is that for macrocycles, we do essentially exactly as well as we did with non-macrocycles. So now fully randomizing structures, and you'll see this procedure on the left here is literally exactly the same for non-macrocycles and macrocycles. No differences. You don't need to think about it. You don't need to worry about it. You just run it. And you get, in the case of HCD protease, perfect results for the top two pose families, actually over 90% for actually perfect for the top one pose family. In fact, for base, we're up over 90% for the top two pose families. Similarly, good results for HSP 90 and factor 11A. Again, now for the conformer search aspect, we're not seeing that conformer search is a bottleneck. Roughly 92% of the time, the conformer pool's input that come to Surflex-Dock from the ForceGen approach are good enough. There's something in there that is close to the right answer. But again, 85% correct roughly across the 128 ligands for the macrocycle set. So this is much, much, much better than people have shown even for cognate redocking of macrocycles. So this really is a completely new sort of result in terms of how well we can do as a field against these kinds of challenging molecules. One thing that Anne mentioned that I want to touch back on, which we brought up actually way back in 2015 with this ligand of 2XNB, is that in the crystal structure, there's actually two alternates that were fit to the density. And in particular, both down here where this little, I think it's a thiazole binds, you see two flips over here. You also see some variation in the amine-containing moiety that is extending towards solvent, which you would expect. Since 2015, we managed to develop an approach called XGen to fit conformational ensembles into X-ray density. And what we see is that when you actually fit a conformer ensemble in here, instead of, these are two conformers each with atom-specific B factors where the bright atoms are thought to move more thermally, or that's how they're modeled anyway. we've been able to show that it's more accurate in terms of modeling the density and a better model actually theoretically of how ligands bind by looking at conformer ensembles fit into X-ray density. So Anne showed you that we actually reproduce both the left and the right flip down here, and obviously we get a lot of variation along the solvent front of this ligand. The same thing is true, and we published a number of papers with our colleagues at Merck on macrocyclic ligands fit into density and how they vary. One very common thing is that you see variation along alkyl linkers. Okay, so over in the backside here, this is modeled in the X-ray structure as these atoms have high B factors, and the nose over here on the left side has a high B factor. What we see is conformational variations in those places. In parts of the protein where the ligand is actually binding well, we see really no variation at all. In both cases, though, what we see is that the docking largely recapitulates what we see in the X-ray density. And remember, in the docking over here, we're using not the cognate structure of the bound protein. It's not the protein conformation that prefers to see these. So we see some additional variation as well. But this is an important theoretical point. We think that people need to absolutely stop thinking about, well, what's the best pose? What's the single pose? What are the coordinates of the thing that binds? It's ensembles. Everything is ensembles thermodynamically. Everything's ensembles in actual crystal structures. And thinking about it this way tells you something about whether you can rigidify what you can do with a ligand. There are cases where standard force gen search is not enough. So tractable macrocycles, so if we look at total molecular flexibility of about 21 to 23 rotatable bonds inside and outside of the macrocycle, those things work just like regular molecules. When we get into FKBP and its ligands, what we see are things like rapamycin, obviously. Rapamycin has a total molecular flexibility of 31. And what we see when we get that large is that the force gen search starts to have a little bit of a hard time. We have developed a deeper search that's adaptive. So for more complicated molecules, it generates larger conformer pools. And what it does is actually rescue the FKBP target. So there's 12 ligands over here, 12 macrocycles. And what we were getting was roughly 60% correct in terms of the top two pose families when we were using normal force gen search. When we actually do the deep search over here, we get all of them right. So in particular for rapamycin, what we see is that the piperacolic acid part of rapamycin binding deeply into FKBP, we get that part right, but then we start to have some deviations from the green crystal structure to the cyan predicted top scoring pose. But we get it exactly right when we do the deep conformer search. Okay, we also did comparisons to autodock variants Vina and Nina. And we normally don't like to run other people's dockers, but it turns out that it's very hard in terms of the licensing regime that exists in the world today to get people to who are professionals at running other docking workflows to run them and be able to publish the results. And we can talk about that a little bit later on. But we worked very, very hard, and Hamani Tandon worked very, very hard, in particular, on pushing Vina and Nina, actually Vina in particular, to the wall on all of these cases. And what we see for Surflex-Dock, we're drawing green lines here at this top two post family 2 angstrom threshold, is that Vina does much, much worse. Okay, roughly 40% correct instead of close to 80% correct for Surflex-Dock. Nina does a little bit better. Long story about this being influenced by knowledge of prior known structures, so that's part of this. But it's doing 60% versus our roughly 80%. For macrocycles, the story is very grim. The folks with Vina published a paper that we cite in our paper on docking macrocycles. So Vina is supposed to be able to deal with macrocycles using the procedure that we used, but it only gets about 25% correct. Nina about 33% correct compared to roughly 80% for Surflex-Dock. If you give Vena the right answer as input, start from the right conformation, it makes a huge difference. So what we think is happening with Vena here is a hard docking failure involving a conformational search inadequacy for macrocycles. We did the same thing for temporal. It makes a very small difference, like a 5 percentage point improvement. If you give Vena the right answer over here for macrocycles, it makes almost a 30-point improvement. Okay, so now the question, when you get to a lot of people are working on really, really, really big macrocycles, okay? This is a paper that we published with Bristol Myers Squibb, starting from a peptidream hit, a 15 mer, and getting all the way over to a clinical candidate, which is not this compound, but you can read about it in the paper. In this case, what we need when we're getting into things of this size, even deep force gen search isn't always able to get these things right. So NMR data can be used to restrain conformational search. So for a compound like this, we get a nice tight ensemble that is in solution. Turns out that this matches very well with how it binds to PDL1. We can use the backbone here now in order to guide conformer search for candidates. So here's a proposed analog. We can restrain the search. We can do docking. And actually, we show that you can get the right answer an awful lot with respect to these kinds of compounds, even at this size. And we can predict then a bound pose. And we can predict things like strain and intermolecular energy. So we showed that this can be very useful for lead optimization. So conclusions. Basically, Fortune plus Surflex-Dock actually works. It's automatic. You don't have to think a lot. You don't have to tweak parameters. You don't have to provide the right answer. You don't have to know the right answer in order to get the right answer roughly 80% of the time looking at the top two poses that are predicted, pose families. It's automatic, it's deterministic. Everything's really different about Surflex in terms of the scoring function, the search methodology, the way we make use of prior ligands, and the automation. All of that are novel features. And in terms of performance, I think that we've been pretty clear about that. We don't know of any other extensively benchmarked method for non-cognate docking that does as well. Part of it is being able to make use of prior knowledge, but a lot of it is just really, really effective search, both at the conformational level and in terms of the docking. So yeah, so that's pretty much it. I wanted to thank our colleagues, Hamani we mentioned, Andrew we mentioned, but also Marietta and Arena, who have worked on the Pine Wall GUI, Matt Siegel, the CEO of Optibrium. I want to thank our friends at Bristol Myers Squibb, Merck, Apertor, and Corteva, all of whom have worked with us on macrocycles in particular and NMR as well. Okay, so now we have some time for questions, and let me see what the questions are in the little chat here. We'll leave it on this slide here. Okay, we have a question. Could you please elaborate how does Surflex-Dock work when there might be just a single crystal structure of the target protein with no information of known ligands? The answer is you can actually look at the paper and see we actually do that control in there. We do actually quite well in terms of when you just have a single structure and you don't have any known ligands. It's more challenging. We really do recommend where possible to use ensembles of proteins. We have colleagues out there, particularly in pharma, who have been using MD to explore conformations if they have a single crystal structure, and then we sample from there and do ensemble docking into protein conformations that were generated with MD. So that's one way to get around it a little bit. We wish we could publish a paper on that, but we haven't been able to get one of our colleagues to cough up data like that. Okay. So let me go to some of the other questions here. Okay, so let's see. You show comparative results for Vena and Nina, which are open source methods, but you don't show results for Gold, Glide, FlexX, or other commercial methods. How well do those approaches work? That is a really good question, and it turns out that there's no way to answer that. So the commercial folks have made it licensing requirements for most people. It makes it impossible for them to publish benchmarking results. So we make our data sets available so that people can run those methods, and we encourage people to make use of the data sets and run the methods, but we can't we haven't been successful in getting people to publicize their results on these kinds of challenging benchmarks. We have some anecdotal data from people that I can't talk about. Let's just say we're very confident in the quality of our results. Okay, we have another question. Have you looked at diffusion learning methods for docking like DiffDock? The answer is yes, we have. I don't want to spoil the fun. We have a paper coming out hopefully in the next few days showing that DiffDock's results in docking are completely artifactual, based entirely on memorization. So hopefully you'll see that very, very soon. We have time for, I think, a couple more questions. Okay. Okay, do the methods offer a way to perform induced fit docking where the protein atoms actually move? This is a question that a lot of people actually ask. And it turns out Ann and I have worked on this for a really long time, over 20 years now, looking at conformational variation in proteins. And what we found is that it's better to use the kind of softer interpenetration term that we have with the scoring function and allow a little bit more bending of the ligand with respect to the force field than it is to push around protein atoms. Because it turns out you can only push them around a little bit. You never are able to predict a movement like antagonist to agonist kind of changes, say, in a transcription factor like estrogen receptor or something like that. Those are the movements you want to be able to predict. The tiny movements that you get from induced fit approaches are well taken care of by allowing your ligands to deform a little bit instead. Let's see, we have time for, I think, one more question. This is one that we get frequently about covalent ligands and structures. We have not done a lot of work with covalent ligands, but there are ways to do it where you chop out a piece of the protein active site where the covalent bond is happening, and you create a positional restraint for your covalent ligands that matches that chunk that would make the chemistry work. And then you just do a docking with Surflex-Dock in the normal way, but using a positional restraint to enforce the geometry that the covalent bond would enforce. And we've shown that that works for a couple of colleagues and collaborators, but it's not an emphasis that we actually have. And
Yes,
I think
and I'd
at this
like
point,
to, there's another question too, about using
Mhm.
AI predicted protein structures for docking. And I think that in general, structures, for example, from AlphaFold 3 are not yet to the accuracy level of crystallographic structures. So for now, we don't believe that AI generated structures can be used for accurate docking.
Ann's 100% right about that. There's just no way you can use those structures for docking for the kinds of things that you want to do.
Great, thank you both, and thank you everyone for watching, for all of your questions. As AJ mentioned, we've run out of time now, but we'd love you to join our next webinar, which is on Thursday, the 5th of December, and we'll be on mastering multi-parameter optimization, developing advanced MPO strategies, and it'll be our Chief Scientific Officer, Ed Chapness, who will be leading that one. So yeah, thank you everyone for joining. Thanks AJ and Anne for a wonderful talk, and hopefully see you all next time.
Thank you, everybody.
Thank you.